In question one, we are asked the form of the gene that is always expressed if present, and that is the dominant form. Number two, which of Mendel's laws best explains why an organism with a heterozygous genotype will show a dominant phenotype? And the answer is A, the law of dominance. So, what Mendel did was he created tall plants, which we now know would have two dominant alleles. Remember, the word allele refers to alternate forms of a gene for a given trait. So when Mendel crossed tall plants with short plants, he noticed that he kept getting tall plants. But it wasn't until, so we call this the F1 generation, all of them were tall. But it wasn't until he did a second cross where he took two plants from the F1 generation, namely two heterozygous plants, and when he crossed them, lo and behold, the short plant showed up again. So what he concluded was that the gene for the short plant had to have been present in the first F1 generation, but it wasn't expressed. That led him to realize that there are alternate forms of the gene, one dominant and one recessive. And that's where he came up with the law of dominance. And if the dominant allele is present, it will be expressed. It doesn't matter if the other allele is dominant or the other allele is recessive. The dominant allele, if present, will be expressed. For number three, we are told that two parents have the genotype heterozygous G, capital G, small g, for a genetic disorder caused by the recessive allele. What is the chance that any of the children will inherit the disorder? So remember, the Punnett squares have the gametes, potential gametes, along the top and the side. So if this is the parent, during meiosis, when gametes are formed, one of these alleles is going to go to one gamete and the other allele is going to go to the other gamete. This is actually the law of segregation. So each gamete possesses only one allele. So these are the potential gametes that a heterozygous parent would form. So if we have two of those parents, then when the gametes unite and form the zygote, then the potential combinations are as follows. And it looks like only 25% of them would be recessive, would be homozygous recessive, and therefore would show the disorder. So the answer to number three is 25%. Question four, which of Mendel's laws states that an organism inherits two copies of a gene and donate one copy of each to their offspring? Okay, so here we're talking about the law of segregation that we just reviewed. All right, here's the organism with two copies of a gene, two alleles. However, when it talks about donating one copy of each, they're talking about the gametes. And it is only going to be one allele per gamete for any given trait. Next, we are told that in pigs, the rough allele, capital R, is dominant to the soft fine coat, small r. If a rough boar and a soft sow mate, they produce several litters. 50 are rough, 50% are smooth. What must be the genotypes of the parents? Okay, so when they say that a rough coat boar, we don't know if that is RR or R small r, because either way, the phenotype would be rough. If they are crossed with a recessive sow, we would get this for our Punnett square. And we see that condition 2 produces 50% soft. So it has to be that the parents are heterozygous and homozygous recessive. Heterozygous, homozygous recessive. Can't be homozygous dominant. Otherwise, you would not have the recessive fine coat up here. For number 6, we have 2 parents with dominant phenotypes. Okay, so again, we don't know if they're have 2 dominant alleles or if they're heterozygous. If they produce an offspring that's recessive, then you have to have one parent, you have to have both parents heterozygous. In other words, if we had one dominant parent and the other one heterozygous, the capital R would be present in each of these combinations. Therefore, you would not see the recessive phenotype. The only way for the recessive phenotype to show up is that both parents, although their phenotype is dominant, their genotype is heterozygous, and it is this lower right quadrant that would produce the recessive phenotype. So for number 6, it's choice E. Next, we are given a dihybrid cross for a plant. Capital P, small p, capital S, small s. What is the predicted phenotypic ratios? So here, the key is to determine the gamete combinations. So the way you do this is you FOIL this, similar to how we multiply 2 binomials in algebra. Okay, so we're going to do first times first. That's capital P, capital S. That's one gamete. Then we're going to do outside, capital P, small s. Then we're going to do inside, small p, capital S. And then finally, we're going to do last, small p, small s. Okay, and then we're going to have the same exact gamete combinations here. And when I make these recessive letters, I try to make them much smaller than the dominant letters and vice versa. It's so easy to get confused. And then you just form the combinations. I'm not going to do that on this video, but when you do, you end up with the 9-3-3-1 phenotype. And it might be helpful to go ahead and memorize that. Next, we're told that there's an American curl cat that has curly ears. The allele for curly ears is dominant, so E would be curly ears. Suppose a cat with a heterozygous genotype is bred to a female with homozygous recessive. So heterozygous would be like so, and a recessive would only have the small e. So if we did our Punnett square, we would have this genotype. Draw the Punnett square, write the phenotypic ratio of the offspring. So it's going to be 1:1, curly to non-curly. And if they produce 6 kittens, how many would have curly ears? Well, it would be 6 divided by 2, since 50% of them would be curly, so the answer would be 3 kittens. Next, we are told that in humans, widow peak is dominant to a straight hairline, and long eyelashes are dominant to short. Okay, if we have a homozygous widow peak man, so that's going to be capital H, who is heterozygous for lashes, so let's call that capital L, small l, is crossed with a straight hairline, so that would be recessive for both alleles, short eyelash woman. So again, that's going to be recessive. Okay, so we're going to do the cross. We're going to determine the gametes. Again, we're going to FOIL this. So on top, we're going to have first times first, that's capital H, capital L. Outside, that's capital H times small l. Inside, that's going to be capital H, capital L, and then last, capital H, small l. And here, for this one, no matter how you FOIL that, you're always going to just get H and L. So that's the only one we're going to do. So our combinations are going to be capital H, small h, capital L, small l, capital H, small h, two small l's, capital H, small h, capital L, small l, and finally capital H, small h, two l's. So it looks like 50% are going to be like this. And then the other 50% are going to be like that. Okay, let's see what they're asking for in this question. List the genotypes and complete the cross. Okay, we did that. And list the phenotypic ratio of the offspring once you have completed the cross. Okay, so it's going to be 50% widow peak long eyelashes and 50% widow peak small eyelashes or short eyelashes. Next, we are asked to perform the following dihybrid crosses and determine the probability of the given offspring. Okay, folks, there's two ways to do this. So let's do it the way we did the most recent one. We're going to take each of these and we're going to FOIL them, and we're going to create potential combination of gametes for both traits. They don't tell us what they are, but that's okay. We don't need them. Okay, so if we FOIL this, we're going to do capital A times capital B. That's first. No, that's wrong. Sorry. First times first is going to be capital A times capital B. Then we're going to do the outside, capital A times capital B. Then we're going to do the inside, which is small a, capital B. And finally, we're going to do the last, small a, capital B. Okay, for the second one, down the vertical side of the Punnett square, we're going to do first times first, that's small a, capital B. Outside, small a, small b. Inside, small a, capital B. And then last, small a, small b. Okay, so what do we notice here, folks? We notice that we're duplicating. We have AB twice, and then we have AB, small a, capital B twice. Same thing here. We have duplication going on. So what we can do here is we can make this easier. We're going to take one of these away, and then we're going to take one of these away. And we're going to make our life easier by doing so. Okay, so now we're going to get the combinations. We have capital A, small a, double capital B. Capital A, small a, big, small, double small, double capital, double small, capital, small. So let's see which one they wanted. They wanted small, small, big, big. Small, small, big, big. So it's going to be 25% is your answer. Now, folks, there's another way to do this. And the way to do this is just focus on the A's and B's separately. So what we're going to do here is we're not going to FOIL. Okay, we're going to make two separate Punnett squares. So first, we're just going to focus on the A's. Alexa, cancel. We're just going to focus on the A's. And over here, we're just going to focus on the B's. Okay, so in other words, for our first Punnett square, we just focused on these two items, capital A, small a, two small a's. And for the second one, we are just focusing on the b's over here. Big B, big B, big B, small b. So now let's go ahead and get our combinations. We have A, 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 A. Big, 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 small, big, 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 small. Okay, so now that we have that, we're going to ask ourselves, what's the probability that we get this combination? So first we would need double A. So double A right here is going to be 50%, or 1 out of 2. Let's do this computation in orange. So there's going to be a 50% chance, 2 out of 4, and if you reduce that, that's 1 over 2. And here they want double capital B. So again, it's going to be 2 out of 4 or 1 over 2. So folks, in math, probability is, what's the probability of getting this and that? We just multiply them. So the probability of getting both of those is 1/2 times 1/2, otherwise known as 1/4, and that's the same answer we got by doing the longer method with foiling and creating the combination of gametes. We got the same exact answer. Next, we are given this cross where 3 traits are involved. So folks, we are not going to set up Punnett squares with all of these combinations of the gametes on the vertical and horizontal. We're going to use the shortcut method. We're going to investigate one trait at a time. So let's start with the A trait. It looks like we have heterozygous times, or crossed with homozygous recessive. For the B, we have same thing, heterozygous crossed with homozygous recessive. And then finally, for the C, we have homozygous dominant and homozygous recessive. Okay, let's go ahead and get our combinations of the progeny. Looks like we have 50% heterozygous, 50% recessive for the first two, the A and the B. And for this, these are all going to be the same. Okay, so let's look at what they want. They want heterozygous for A and heterozygous for B. So that's going to be 2 out of 4 or 1 out of 2. That's going to be 2 out of 4, 1 out of 2. This is going to be 4 over 4, otherwise known as 1. So we're going to multiply 1/2 times 1/2 times 1, and we're going to get 1/4, or 25%, as our probability to get that as the offspring. In our third and final problem, it's very similar to the one previous. We have heterozygous for the A. We have heterozygous for the B. And we have heterozygous for the C. Okay. What are the chances that we're going to get recessive for all of them? Okay, we already know that when you cross 2 heterozygous, the chance of the recessive is just 1/4. But we'll do the first one. Now let's do them all. Okay, So the chance of recessive for the A is 1 out of 4, for the B is 1 out of 4, and for C is 1 out of 4. So that's 1 over 4 to the third power. We use the power rule. That gives us 1 to the third over 4 to the third, 1 to any power, sorry. We use the power rule, 1 to the third. When you do the power rule, you do not multiply that exponent times the 1. You multiply it by the exponent. So it's 3 times 1, 3 times 1. You get 1 to the third over 4 to the third. 1 to any power is 1. 4 to the third power is 64. And if you put that in a calculator, you'll get 0.0156. Now, to convert from a decimal to a percent, let's do that up top in orange. So 0.0156 equals X percent. We're going to take away the percent sign and replace that with division by 100. We do that because the word percent, per cent. Cent in Latin is 100. Per is fraction line. So we're going to get that. We're going to multiply both sides by 100. And when you multiply the left side by 100, that's tantamount to moving the decimal places 2 to the right. And you're going to get a final answer of 1.56% chance to get all recessive in that cross. Next, we have a string of multiple choice questions that don't require math. Number 11, for an XX female to express recessive sex-linked trait, she must have 2 recessive alleles. If one were dominant, it would manifest. Number 12, human height is a continuous range because it is affected by the interaction of many genes. Many is poly, genes, polygenetic. Choice C, red hair and freckles are commonly inherited together. They are A, linked genes. Which pair in the diagram are most likely to be inherited together? Don't know if you could see that, but it's going to be A and B because their loci are close together. In a newly discovered flower, the color is determined by 2 different genes. Again, that's polygenic inheritance. What is the main reason that sex-linked disorders are most often observed in males? It's because the male only has one X chromosome. So the Y chromosome can't mask it. The female is going to have 2 X chromosomes, and there's more of a chance that it gets masked because you could have the dominant allele on the other X. Most sex-linked disorders are recessive. Finally, which are considered homologous? You won't be able to see this, but here there's capital A, small b, capital B. In other words, the loci match up for these 2 homologous chromosomes. Now remember, the word homologous means not identical. Homologous means they have the same foci, but they're not identical. Identical chromosomes occur during replication, where you have an original chromosome. Okay, so these are homologous, similar shape. They carry genes for similar traits, but they could be different alleles, alternate forms. That's homologous. However, during mitosis or meiosis for that matter, when these chromosomes duplicate or replicate, they are going to be identical sister chromatids. So that's the difference between the word homologous and identical sister chromatids. Next, we have a man with type A blood marries a woman with type AB. Based on what you know about blood types, what is the only blood type not possible in any of their children? Okay, so a type A male could be one of two genetic or one of two genotypes. He could have one allele with the type A antigen and the other allele with no antigen, or the type A father could have two antigens that are capital A. In terms of the mom, who's AB, she's going to have one of each. The allele for antigen A and antigen B are considered co-dominant. Okay, so it's going to be one of these two scenarios. So here we're going to have two A antigens, so that child would be type A. Here we're going to have an A and a B, so that child would be type AB. Here we're going to have an A and an I, again type A. And here a B and an I, that would be type B. Over here in this scenario, we're going to have a type A child, a type AB child, a type A child, and another type AB child. So the only one missing is double I, therefore the child will not be type O.